Hello, my name is Lea Bennett. I'm an assistant professor at the University of Oklahoma Health Sciences Center in the Department of Ophthalmology located at D. McGee Eye Institute. I study inherited retinal diseases. These diseases occur because of a genetic mutation causing loss of vision and blindness. There are approximately 1 per 3,000 individuals afflicted with an IRD. There are no effective treatments to restore vision for these patients. IRDs typically begin during childhood, which has lifelong implications for both the child and their family members. The economic burden associated with IRD is $31.7 billion in the United States. This includes both economic and well-being costs. People living with an IRD often have other comorbidities, such as an increased risk of developing mental health conditions like anxiety or depression. Patients with IRD are 30% less likely to be employed than the normal-sighted population, which leads to society-wide losses from reduced taxation revenue due to decreased workforce participation and higher government expenditures. Research into current and emerging cost-effective therapies and interventions is desperately needed given the substantial economic burden faced by those living with vision loss. The research goals of my laboratory are to understand the effects of genetic mutations on the retina and to investigate novel therapeutic directions that could be beneficial to patients with IRD. We use clinical and genetic testing to associate gene mutations with the patient's clinical features. There are around 300 different genes associated with IRD, with many overlaps in both the clinical diagnosis and the genes associated with IRD. For example, patients with a mutation in the gene called PRPH2 can have widely different clinical diagnosis. In fact, PRPH2 has been associated with retinitis pigmentosa, shown in the central panels. Retinitis pigmentosa is an IRD that's initially characterized by loss of night vision as well as loss of peripheral vision. This is because rod photoreceptors are diseased. However, sometimes a mutation in PRPH2 will cause macular disease, which is shown in the right panels. This results in a loss of central vision because the cone photoreceptors are diseased. The white flecks in the bottom right image is similar to what we find in patients with Stargardt's disease, which is an IRD caused by a completely separate gene called ABCA4. So you can see how the overlapping genes and clinical characteristic lead to uncertainty and often an inaccurate diagnosis. Because genetic mutations associated with IRD primarily affect rod and or cone photoreceptors, we use different clinical assessments to evaluate both rod and cone function separately and perform extensive retinal imaging to study how the structure and function of the retina is affected by the genetic mutation. We work to resolve inconclusive genetic testing results. In the past, patients with an IRD were either misdiagnosed or given a devastating prognosis of becoming blind with no hope of treatments. Within the last decade, information regarding genetics responsible for disease and the increased dissemination of knowledge gained about clinical features of IRD has significantly changed the standard of care for these patients. As recently as 5 years ago, patients had to wait up to a year before they would receive the results from their genetic testing. With advances in genetic sequencing technology, we can now run a comprehensive panel of all currently known genes associated with IRD in a short time, with patients receiving the results from the genetic testing in less than 1 month. However, only 50 to 60% of patients have an identifiable mutation in one of the known disease-causing genes, leaving many patients without a conclusive genetic diagnosis and a gap in our understanding of IRD mechanisms. Although a known mutation may not be found, new DNA mutations in IRD genes are often identified. New DNA variants are classified as variants of unknown significance. To decide whether these variants are pathogenic or responsible for the patient's disease, it has to segregate with the disease. This means that the variant will be found in affected family members or other individuals with similar disease, but not found in unaffected family members or the general population. Additionally, functional assays that test the effect of the mutation in cell-based assays or in animal models can provide the support required to reclassify the mutation. This is vital so that clinicians can determine the accurate clinical diagnosis for IRD patients. Together with the rapid turnaround in genetic testing and advanced sequencing technology, detection of variants of unknown significance has increased and created a bottleneck in classifying these variants, which delays the genetic diagnosis. The multidisciplinary process of genetic testing and variant classification can take years to coordinate and resolve. We are working towards quickening the diagnostic process by developing standardized assays to interpret these variants. The variants are identified through genetic testing and subjected to an in silico analysis, which just means computer modeling. This determines the cellular defect predicted to occur with the DNA mutation. The prediction is then tested in a functional assay in the laboratory to provide evidence to whether the variant is pathogenic and causative for the patient's clinical diagnosis. We also want to understand the mechanisms of disease associated with these genetic mutations that cause IRD. Some genes associated with IRD cannot be studied using the traditional animal or cell-based models because they do not replicate what is observed in the human retina. This is because of inherent complexity of the human retina and the natural differences between species. Recent advances in stem cell technology allow us to overcome these limitations. Adult cells such as blood, skin, or urine can be used and reprogrammed to induce pluripotent stem cells, or iPSCs. These cells are essentially a blank slate and can be differentiated into any cells in the human body and directed to develop into 3D tissue-like spheroids. The ultimate goal is to use these cells from a diseased patient, correct the defect in the lab, and put them back into the human donor in order to treat the disease. We use iPSCs from human donors and direct them to form retinas in a dish, or retinal organoids. These 3D tissues contain multiple retinal cell types, including photoreceptors. We use these iPSCs from patients to create retinal organoids that contain the genetic mutation, and we use them as a model of the human retina to study disease mechanisms and for finding therapies for these patients. There is a critical need for increased research funds so that we can identify the remaining unknown causal genes, continue research to develop treatments and therapies, improve access to genetic testing and counseling, and to promote care and treatment and ongoing education of all professionals involved in eye care for patients living with an IRD. Thank you for your interest in vision research. I'm especially grateful for the opportunity to participate in the 7th Annual Emerging Vision Scientist Program. Thank you.